up and running? Very good. Did you just get my slides up for 1 second? Here we are. Cool. Does everyone see that okay?
Yes, we can see that. Thanks.
Cool. So,
Yes.
Thanks everyone for joining again. And this is day one of our protein engineering series of workshops. And before launching into any of the technical details, I think Pearlie gave a really nice introduction just a moment ago, but I can elaborate a little bit on that. So again, I have a fair amount of expertise in this particular field of how you design proteins, manufacture proteins for a variety of uses, in particular with a medical focus. But certainly was interested to hear earlier some of your kind of comments and feedbacks about other areas this intersects with where you can design and make proteins. And I won't give kind of the full backstory here right now, but just to say that if you stick around for the third day of this workshop series, it'll be kind of I think, a nice opportunity where you can ask questions about career. I'll go a little bit more into kind of my own journey in science and how, I went between continents to get different degrees, like my PhD at Cambridge, and then even the pathway that I've been starting relatively recently about starting up a company, being in the biotechnology industry. So kind of like a little bit of a teaser, so to speak, to stick around for all three of the lecture series, and then you can learn about that, kind of ask questions of what it's like, and then we can engage more there. So just as a kind of overview of how I'm planning to piece together this series of workshops, today, day one, I kind of have it under the title of Designing Your Proteins. So how to identify proteins for your projects, work with their sequences. Day two is once you have a protein designed, let's say you have a wonderful protein that's going to solve your research challenge or make a difference in the world. Then the question is, how do you actually produce it? And there's a lot of kind of important concepts there that influence protein design and how proteins work kind of in a laboratory environment. And then after putting those two series together, it's then that question and answers about the workshop itself or careers. Day three is really all about an opportunity for you to learn more, ask questions, and just have a nice chat, hopefully, about science and biotechnology. So starting off with today, day one, I think I'll be roughly dividing this in half with kind of a little bit of a short break in between. The first half is covering over some of the concepts that you'll need before you start launching into protein engineering yourself or any of these protein design concepts. So we'll start there. And then once we've gone over those concepts, I think I want to devote pretty much half of this time, so quite a lot of it, to really, getting our hands involved in this process, seeing what it's like, using some common tools that, even myself in my own research often use to look at proteins, design their structure, and kind of imagine that in a couple use case examples. And then at the end, as part of this workshop series, we also have a little short assignment to help you kind of practice those practical steps. So hopefully the idea is, once you see all of that and done your practice, then in the future, if you need to use these tools, you'll kind of have all the basics you need to get up and running. So kind of the title of this slide, to start out with is why focus on proteins? And I really like this image here because I think it answers it in the picture, which is this is an illustration that was made of what the inside of a cell looks like. And you can see kind of this, a beautiful zoo of different proteins and all these different colors, some forming these tubular structures, some forming things on membrane surfaces, enzymes, machinery of the cell. And I think what really is important to stand out here is as you look at all this complexity and everything that's happening inside of your cells, it's almost entirely the proteins that are doing that work. So like I've heard a good metaphor before of, your cells are almost like, protein robots where the proteins are the machinery, the kind of cogs in them that function, catalyze reactions, make life possible, put together. And so if you're able to design and make proteins, then you really have an ability to kind of master this key area of technology where you can, make nano machines, make things that interface with biology, like new medicines, new particles. And so, hopefully you should feel quite empowered after this workshop of, if you can work with proteins and make proteins, you can do quite a lot. And I think we addressed this a little bit for those of you who are there at the very start with a kind of short icebreaker activity, but kind of thinking about what is protein engineering and what are some of the concepts that we'll learn today. I think one of the things that maybe I'll just briefly highlight here, beyond what we went over during the icebreaker activity at the start, was, don't think of proteins as being too abstract. I'd bet right now where you're sitting, there's a good chance that there's a protein-based invention or piece of technology that you're using, in this very room or while you're sitting at this. It could be like a wool shirt or, your leather shoes or glues that are made of proteins, medicines that you might be using, things in your diet, how enzymes are used in the food business and how the food itself is made of proteins. They're really quite fundamental and pervade your life, not just as research tools that say, companies like mine would be working on, but, really things that you can build out of, you can consume, you can even make energy out of them in some cases. So, I think there's a real power when you realize just all the versatility proteins can have. Protein engineering itself, I can at least take my own turn, since some of you did your own exercise of defining this, but I can take my own turn at this. And, when I think of protein engineering, it's really thinking about, again, these proteins as machines. And so, in conventional engineering, you might, figure out how the machine functions, how its parts work. Protein engineering is a similar concept, understanding how the different elements of a protein fit together and then allow it to give the function that it has. So you could improve its function, you could fix its function if it goes wrong, say in a disease, or, create new kinds of machines for new functions. And I think I hit on this a little bit, but, in the biotechnology industry especially, since I know some of you are quite keen in learning more about, say, my experience there and what it's like in biotech, proteins are really quite central. I have just like a little collage of some examples, but there's, ways you can replace harmful dyes with proteins. You can even mine metals with proteins that break up rocks, making next generation biofuels, sustainable foods, and then a whole bunch of things on the medical side of, treating rare diseases with proteins. Antibodies, of course, is a huge area that's been used to treat now more than 100 diseases across the world. Vaccines, gene therapies, areas like this. What they all have in common is they rely on these protein nanomachines to work. And again, if you can make these protein nanomachines and you can design them, then I think you can pick one of these areas or other areas that interest you and, really think how you could use this to make a difference. Now, to get a little bit more on the technical side, I think it's worth kind of breaking down this big word of protein engineering into a few examples. And I kind of have them as a sort of hierarchy of complexity. So the first one that maybe some of you have been done already in your research is like small mutations or adaptations to a protein sequence. So, examples might be kind of like point mutations on an enzyme, modifying active sites, but, kind of like in this diagram here. You can maybe get a laser pointer going just to make that a little bit more obvious, but, it's like, imagine you have this whole big protein and you're just changing this one little small region of it, but, that can have a very big influence on its function. So it still can count in protein engineering. How do you decide that best modification to make and where to make it? And the next level of complexity up that's also within protein engineering is this concept of fusions. And so this is, I think, a really powerful concept in biology where you can treat proteins in many cases as being modular. So a famous example, and the one that I've illustrated here, is, this protein called GFP from jellyfish that glows green. You can treat it as almost like a module that you can stick onto other kinds of proteins. And then, if this yellow protein here has a particular function, maybe it binds a particular receptor, then you can fuse it onto this GFP protein. And now you have something that binds to that receptor and then glows after, upon binding. And so it's, almost like a biosensor at that point. So I think fusion's a really powerful area where you can mix and match functionalities and you really, build things out of different parts. One layer up is these computer-based methods that can redesign protein sequences. So the most famous example of these are little binding proteins, where you'll have a scaffold and then a computer algorithm will figure out kind of all the tweaks to the amino acid chains of that protein that might make it have, say, stronger binding or increased catalytic activity, but, kind of just having a rough scaffold in place, and then the computer tries to optimize on that scaffold. And I'll go into a little bit more detail kind of how that looks in a second, but just kind of as a concept to introduce. And then the most recent, and I think one of the more advanced areas in this field that's been growing recently, is the idea of proteins that are kind of totally synthetic, that never existed in nature. There's this really wonderful illustration here where you have this kind of blue sea and a few yellow islands. And what it's trying to represent is, all of the proteins that we have in nature across all of our species really are just a tiny fraction of all the possible proteins that could exist if you just string amino acids together in different ways. And so there's a lot of untapped potential, which is kind of this vast blue sea here, that are proteins totally unlike anything that exists on Earth at the moment, but that you could imagine designing and then having function on. And so kind of there's this broad scope, across all these different areas that fit under protein engineering. And, we can see from those examples that depending on your problem, depending on what you're looking at, you have lots of options in that tool belt. Just to kind of frame what we'll be talking about today, this workshop series will kind of cover these first two bits, and you'll see them in the practical demonstration. So you can look forward to how some of the concepts around mutation work and how some of the concepts around protein fusions work. And then I'll have some bonus slides that I can distribute after the workshop that will give you a more extensive peek into some of these other areas, as well as other useful details to help you along the way. I think the last note that I want to make before we kind of jump into some of the more practical learning parts is that protein engineering, as I'm phrasing it here, is as much of an art really as a science. And what I mean by that is, as you'll see in the demonstration, there's not always a very hard, clear rule of, I should do exactly this, or this is, in this situation, here's the only way I can respond. But you'll have many options. There's, I think, a lot of intuition and just kind of built experience that goes into protein design, kind of at least in the categories that we'll be learning about today. And so it can seem a bit scary at first because you might be kind of questioning, am I doing the right thing? Is this correct? Those kind of questions. But, really don't be too afraid by that. Like protein engineering is hard. We kind of fail all the time, but you do your best attempt and, you can use your creativity really in trying to look at these protein structures and figuring out how to improve them. So I think that's all the kind of background framing, just to help put everyone in the mindset about what it's like to design proteins. Now, for these slides, I'll be going a little bit into kind of a reminder so that everyone's hopefully on the same page, same starting point before we go into the demonstration. So for some of you, this might be more review. For some of you, this might have new information, and I hope to kind of sprinkle in some interesting anecdotes throughout too. Just the most basic part of this, which I alluded to already, is proteins are made out of amino acids. So hopefully folks remember this from their biology courses, but there's 20 standard amino acids, at least in humans. And the thing to remember is that each of these will have a unique chemical property. It's like these ones on top with these kind of hydrophobic nonpolar side chains. One of their properties that you'll have useful to remember is they'll want to kind of bury themselves inside the protein's three-dimensional structure. And you can kind of think of why. They don't like water. So water's on the outside. Your cells are always in water. And so these kind of hydrophobic residues are kind of a way that you can turn the protein inward and form these hydrophobic cores. So, already that's kind of a basic concept that you can start to think around using, how that might help you design proteins or, fix a protein that's not functioning properly. And of course, there's the polar amino acids, which are kind of the opposite. They'll tend to dot the surface of a protein and help it be solubilized in water. There's certain amino acids that are electrically charged here on bottom. So depending on the pH, which is, the amount of these protons that are in the solution, you might get these amino acids attach an additional proton, which will make it positively charged, or lose its proton, which will make it negatively charged. And then I'll touch on a little bit, because these can be quite important, these special case amino acids. In some of your bio lectures, they might have been kind of glossed over, but I think when you're thinking of protein engineering, these can be really critical because many of these other amino acids, they might be changed and you'll still get a protein out, but these ones are quite influential. Glycine is fairly neutral. It's quite small and doesn't really have a clear functional category within like these groupings here. Cysteine is very unique in that it can form bonds across your protein chain. So I have a little cartoon of this next. If you imagine your amino acid sequence is a long string of amino acids, cysteine is kind of the one thing that is the exception to that rule, that will actually be able to form these covalent bonds across different parts of your protein and kind of link them together. I think the last special case I wanted to highlight is this amino acid proline. And what's somewhat unique about proline is that it kind of forms these kinks in the structure. Where those will be relevant, I'll mention in a second, but just kind of keep your eye out when you see amino acids like cysteine and proline that, they might be quite important to what you're seeing at the end. Now, where this kind of develops from there is you have your primary amino acid sequence, and then, hopefully this will be a reminder for some folks, but then is kind of developed onto what's called the secondary structure, which we'll talk about next, and then the tertiary structure, which is like your 3D folding. And then also don't forget this concept of what's sometimes called like the quaternary structure, which is totally different amino acid chains that might have to come together and function together for your protein to have its needed activity. Starting with secondary structure, I think particularly if you're just getting introduced to this field, it can be quite a powerful way to start thinking. Whereas Jane Richardson, who kind of pioneered at least our current way of looking at in designing these secondary structure elements. And there's two main categories, as you can see here, these alpha helices and then beta sheets. And I'll have on the next slide a bit of text to kind of help almost as a resource remind you where this can be used. But I think the basic concept is these two simple kind of fold structures really explain a whole lot of this more complex biology of how proteins fold, form their structure. And so if you kind of know these two, and the sort of loops and things that connect them, you're already a far way there to understanding protein folding. So instead of the concepts and the rules, this is just a more detailed view of what those helices and sheets look like in 3 dimensions. I think when I think of the helices, maybe a good parallel is to think of like a rod in everyday life. They can be a bit more stiff and structured if you imagine it kind of moving around in the protein. And these beta sheets, kind of like their name implies, could be, like layers of a book. And just like layers of a book, if there's multiple together, like in this illustration here, they might form like a stronger structure. Or if there's, single sheets in different orientations, they might kind of have less of a firm kind of orientation or position within the protein. And so some of the kind of details to know before we go into our demonstration is that we're already very good at predicting these structures computationally. So, an algorithm, and I'll show you a couple once we go into the demonstration, predicts, there's an alpha helix there. There's a good chance that there's an alpha helix there. It tends to be decently good at predicting. I think another point that's a bit more subtle, but for those even following some of the really revolutionary developments in predicting 3D protein structure, so like tertiary folding, a lot of it really does come down to just, does the algorithm be able to position and orient these secondary structures correctly? And you can kind of see this almost in the drawing earlier. There's the parts not in secondary structure are kind of those gray loops, but really the core of the protein, this particular kind of barrel that she's holding there, is just a well-structured position of, these secondary elements. And there's more advanced ways you can try and figure out, say, the small details of exactly where this is positioned, all of that. But, you get a lot of the way there to understanding your protein, just if you can predict where those elements are. And the other term you might come across that's important in protein design is what are the parts of the sequence that don't fall in these categories? And at least some of them we call like disordered sequences, in that they don't really adopt a permanent structure. They might kind of change how they look and all of this. And of course, have to keep that in mind as we design our proteins. They can be areas that are a bit more unpredictable. If you modify them, it's hard to know what effect they'll have. But just kind of keep those categories in mind. Now, as I alluded to a moment ago, with predicting 3D protein structures, we're really just in the past few years living in kind of a new era for this. So, if you're just starting out your careers here, you should be, quite excited by this. We have tools where you can input amino acid sequences, which is, what these letters are. Each letter is a different amino acid. And then it can predict, oftentimes with fairly good accuracy, what your folded 3D protein looks like. We're going to use one of these tools in our demonstration because, they are kind of a central really to how the field works now. But I want to issue a few kind of caveats and notes to be aware of. Again, it's going to be a bit of text here, but it's partly so this is a resource you can come back to later. To summarize the text, what I'd say is that, first of all, you have to realize that many of these tools were trained on natural sequences that we've, studied in nature and kind of learned their patterns very well. So if you're, say, engineering a protein that looks quite dissimilar to anything in nature, or, deliberately trying to take something natural and then make it look, have an unnatural fold to it, for example, those might be areas where these algorithms could struggle more. Now, as you'll see during the demonstration, most algorithms will give you confidence scores, which is actually part of what you see as the colors here, to help tell you, what's the probability that this is correct of a prediction. But, you should still kind of have that caution as you use some of these tools, and we'll use some of them today. Other kind of main takeaways to have as we use some of these tools is the disordered regions that I mentioned before. Like, it's somewhat funny. Sometimes you can put like two predictions into these algorithms, and then it'll give, like confetti looking structure that, is totally implausible, doesn't look like any real protein. And that can happen if the algorithm gets confused by these disordered regions and just thinks your protein is unfolded or can't figure out how it's folded. So, be quite wary of those. They'll be highlighted usually in our predictions, and I'll show you an example actually during our demonstration. But they can be a bit of a warning sign of, the algorithm struggling to make a prediction. And they also are not currently able to do very small scale effects. So I mentioned at the start, we're going to do two kinds of protein engineering today, like mutations plus fusions. The fusions, it can be quite helpful to, predict, are these two domains assembled correctly and will they fold together? The mutations, though, are not currently covered by these algorithms. So keep that in mind if, you're interested in that mutation function. And then the last caveat that I want to raise, because I think it's one of the most common mistakes that I find people make, is these prediction algorithms take whatever input you give them and give you a prediction. They can't tell you whether the input that you gave them is correct. So what I mean by that, and there's some examples on the slide, is say you handed it the amino acid sequence from a database, but in reality, maybe in the body or in your cell, that amino acid sequence is chopped up naturally or has this modification to it. You do have to kind of understand your protein, understand how it looks in the body, because otherwise, if you give the wrong amino acid sequence, then the algorithm's just going to, give you some output, but it won't be a meaningful output. So, keep those in mind. And again, I'll walk you through how this actually looks in practice in just a few minutes. I think some last notes, just to balance, I guess, some of the positive notes of how much progress we've made with being able to predict structures with a few caveats, is we're used to seeing protein structures, and during today's demonstration, we'll see them as these static structures. But do keep in mind, like this really nice set of illustrations here, that proteins, they move around. They're not just a fixed crystal that doesn't, adopts a single structure. And some proteins will move around more than others. So kind of the gray here is, different motions of this protein. Some were quite extreme, like this little peptide here that can move all about. Others you can tell, follow its structure very closely. But I think it's important to mention to keep in mind of the structure that you're seeing is a snapshot, and it might be an incomplete snapshot or might not cover everything. And so I'll show you how to design around a particular snapshot. But if you're doing this in your research or as a company or, for your academic work, it's worth maybe collecting a few snapshots and comparing them and seeing, do you get a consistent set of answers? That makes sense. And then kind of a last fun little note, but I think it's an important one before we dive into the demonstration, is, remember that proteins exist in this really strange and kind of unfamiliar world that it can be quite easy to oversimplify. And so I have a kind of collection of, at least to me, quite surprising facts of, what it would be like if you were shrunk down to the size of the protein. What would your world be? It's an area where there's basically no gravity. Like gravity is too weak to have effects on most proteins because they're so small. Inertia works very differently in kind of the solutions within cells. Everything is densely packed and, moving as fast as a car. But, a protein's a nanometer long, but it's moving, 100 kilometers an hour in some cases and bumping into other proteins a million times a second. So we simplify, I think, to help us do protein design. And you'll see these, very beautiful structures later of during our demonstration of here's a protein structure. It looks static, it's not moving. But I've, put this as the final slide before we transition to the demonstration to, really help reinforce this notion of, I guess, humility. You can kind of view it in that, we have to be a bit humble designing proteins. It's very difficult work, and we can make our best guesses, but it's hard to get an intuition for how proteins work. And the best protein engineers that I know, they just kind of develop that intuition over time of these hydrophobic cores of proteins, or how different proline kinks will affect secondary structures. And they get that intuition over time. So don't be discouraged if you don't have that click right away for you, because it is very unusual and strange. Cool. So this is now prepping directly for when we're going to go into the demonstration. Basically, the starting point of the demonstration will be looking at amino acid sequences, and then we're going to look at the structures, and then how we can modify both levels, the primary amino acid sequence, and then modifying the structures. So I want to give a little bit of hints before we go into the demonstration. So then hopefully as you see these tools be used, it makes a bit more sense, what we're looking out for, why we're doing what we're doing. So some hints on amino acid. Over on the right is this kind of all-important table of what the amino acids look like and their different chemical properties. Just like I showed an earlier slide, a different version of this, but the hydrophobic amino acids, the charged amino acids, the kind of special ones like cysteine, all grouped together. And then it can be helpful, especially as you're starting out, feel free to print out like a version of this as you're working or doing protein experiments. Because if you have this by your side, you can then ask questions like, in my amino acid sequence, is there a whole bunch of one type of amino acid grouped together? So maybe a big stretch that's really hydrophobic. For example, proteins that can insert themselves into a membrane of a cell, kind of that lipid membrane that cells have if they have enough of these hydrophobic amino acids. Or there's certain charged parts of proteins that can be really poor for their binding or their properties. And so, it can be as simple as just like a bunch of, in this example, D's and E's in a row. And if you just notice that, you've, I think, already gotten insight into your protein and maybe even an idea for how to design it. What if you made that less positively charged or more positively charged? How could that affect the function? Like I said, make note of these special amino acids, particularly in eukaryotes, those cysteines that form these bridges across the amino acid sequence can be things that you don't want to disrupt. So I guess I'll simplify it and say as much as that, or be very careful if you end up changing them. And then kind of these last points here, I won't dwell on too much right now, but I'll have a bunch of bonus slides that I'll share after the presentation that give long lists of motifs. So I give two examples here, but there's many of them, like this NXS or NXT. And you can look through your amino acid sequence, and then, for example, anytime you see the amino acid arginine for N, any amino acid but proline, which is what X means, and then threonine, you'll actually have a sugar residue that tends to be attached there. And so you can already just, doing a quick text search, something very simple, you can learn about, where is there going to be sugars on my protein, or, this protease cleavage site, where might my protein be cut. And the last category that we'll see in the demonstration is kind of overall properties. So we're talking here about properties of amino acids, but then when you put all those amino acids together, what does it make? And there's some really useful numbers like a isoelectric point, stability index that you can use to predict how your protein will function. So again, this is just kind of a preview, and I'll show a bit more concrete examples in a second. And then the last point with tertiary sequences and how proteins fold. I have a nice little animation here to show an example, but remember here we have these ribbons, which is going to be our beta sheets, and then there are helices, which are going to be alpha helices, and then kind of these non-secondary structured regions in between. And so first question you'll want to ask is just how much of it is well-defined in a structure, how much might be disordered, or not clearly in a secondary element. What does it overall look like? So this protein here, the two colors are actually showing a dimer. And so if you're not studying the protein as a dimer, you're kind of studying the wrong thing, so to speak. Your predictions won't hold because the protein only functions in this particular state, for the example I'm showing you to the right. And we'll see a concrete example of this, but already I think it's important once we go into the area of protein fusions to look at a structure and think ahead, what areas might be safe to modify. So on my protein over here, And just on a quick glance, things to look out for are the ends of the amino acid sequence. So if you remember, the start of the amino acids are called the N terminus. The end is called the C terminus, where the protein terminates. And both of those can be really nice connection points. I won't be able to find them. This actually might be one of the termini over here. But, looking at those and where they're positioned can be really good places for protein fusions if they're accessible and not in danger of disrupting part of the protein's function. The other thing to look out for is there, a natural loop or internal position where you can maybe make a fusion or modification. So, just informally, my eyes kind of drawn to these two extreme loops on either side. Those could be areas that we're actually able to modify the protein, make changes. So we'll see a concrete example of this in a second, but hopefully this at least gives you, again, what to look out for as we go through this. Now, this will be kind of a bit of a pause, but before going into the demonstration, I'm going to give a chance. Did anyone have kind of a burning question going into it, or things I can help clarify of the concepts before we actually try kind of putting our hands into doing this? Give a moment or two. I think we're doing well on time so far. And then if not, I can launch right in. Cool. All right, let's do our demonstration then. So for this demonstration, I picked hopefully will be a nice example and a good motivation for both of the projects that we're doing this week in demonstrations. And it's also a bit of a personal story. So during my PhD over in Cambridge, I worked in the lab that actually had the scientist here, Cécile Crosnier, who discovered how the malaria parasite binds onto and infects red blood cells during malarial infections. And that was, of course, a very fundamental discovery of just understanding how this disease works, kind of the cause and origin of it. And since I have some experience with this particular protein, from that past story, I thought it'd be a really nice example that we could work with this week for a particular problem. Because, right, if you're going to be designing a protein, always have in mind, how's this going to help someone in the real world? What's the problem I'm trying to solve? And the problem that I'm going to do for today's demonstration is about making a vaccine for malaria. So in research, from Cécile and part of our lab, is a really nice aspect of if you create antibodies against this Rh5 protein, it has the effect of not just a traditional vaccine of, immunizing against the parasite, but the antibodies will block the entry point that the parasite needs to burrow in and infect red blood cells. And so you can imagine it'd be quite an attractive vaccine for that reason. It has the kind of dual functions of traditional vaccination plus blocking infection. And so it might sound a little bit intimidating, but hopefully I'll kind of walk you through how it's actually not too hard. But the task that I'm going to set out for us in this demonstration is, can we design a kind of next generation malaria vaccine based on this relatively recent discovery just in the past 10 years or so of this Rh5 protein that the parasite uses to infect humans. So again, it can sound quite grand, but hopefully you'll see within half an hour to 45 minutes, I'll walk you through the steps, and hopefully you'll see, it's something that hopefully you feel empowered you could do it yourself and is not actually all that difficult. Now, to explain, I guess, the approach that I'll show to this problem, because there's many ways you could use Rh5 in a vaccine. This is the way that I'll be showing. So it's taking inspiration from, those of you who are following the news, a really important medical breakthrough a few years ago of the first malaria vaccine that was approved, called RTSS. And nicely enough for our course here, RTSS was very much an engineered protein vaccine, just like we're working on today. They took this protein from the malarial parasite called CSP. They fused it onto this protein that basically forms, as you can kind of see in this cartoon here, like these almost virus-looking structures. And because of that structure, it makes it very good at eliciting the immune response. You have this malarial protein presented as if it looks like a virus. So, the immune system generates a much better response than if you just put the protein alone. And this vaccine, you kind of see on the summary on the right from the World Health Organization and BBC, has had really good promise in showing that malaria vaccination is possible. But as you know, if you can read the numbers there, it's clear there's a long way to go. It's, a majority of cases still can't be prevented, even if everyone was vaccinated with this. So the question is, can we use the structure of this engineered vaccine already in use of a malarial protein fused onto this immune response generating protein to do the same thing for Rh5, this protein that's needed for malarial infection into blood cells? So that's our goal that I'm going to set out. And let me talk through the steps before I actually show them so if we can follow through a bit better. The first thing we're going to do is find this parasite's amino acid sequence for the Rh5 protein. I'll show you a really useful database for that. Then we're going to do one of those rational mutations, kind of in that first category of engineering I mentioned, to prevent our vaccine from binding onto blood cells. Remember, Rh5, we like it because it's how the parasite binds onto blood cells. We don't necessarily want our vaccine to have that function. So I'll show you a very simple way that we can actually remove that function, just by rational mutations. And then we'll get into a bit of the more complex parts, but again, it shouldn't be too intimidating, where we're going to make a fusion of that, you remember it's called like the HSBAG, this kind of immune response generating part onto our Rh5. And so we're going to look at the 3D protein structure, use that to inform how we can make our fusion, and then use the software tool called Benchling to kind of piece everything together and make sure that it works, or predict how well it will work. So I think this is my last summary before we jump in, but kind of breaking down the steps in a bit more detail. Thankfully, the organizers have already done a bit of an introduction to how the Benchling tool works, so I won't go too much in detail of how you navigate, but just kind of help orient you there. Then we're going to use the UniProt database to find this parasite protein and the information we need to be able to start engineering with it. We'll load those into Benchling, analyze the amino acid sequence to get some important information before we actually start making our engineered mutations. And then we'll go into the part of making that fusion. So we'll introduce the mutation so it doesn't bind to blood. Then we'll make our fusion, use the AlphaFold algorithm to predict the 3D structure. And I'll show you how you can do it yourself because it's quite easy and accessible tool. Then also show you how you can visualize protein structures using this tool called Molstar. And then we're going to kind of put everything together, make our fusion based on the structure, and we'll have our final vaccine. Cool. So does that sound good for everyone? Actually stop sharing my slides over here, and then that. And we'll transition on to the practical demonstration. Cool. All right. So for this part, I think for those of you watching, the way that I would approach this is kind of watch what I do first, follow the demonstration, and then again, there'll be an assignment given you after this where you can sort of practice it on your own and follow the steps. So, just try and follow what I'm doing, maybe take notes if that's useful for how you tend to learn things best. And then, again, you can put it into practice right after this workshop or, over the next few days through that assignment. So, don't worry too much about, doing things in parallel or trying to do the same step as me at the same time. Because, I will be trying to part of my goal here is I want to show you a lot of tools so you can, do things yourself and really tackle important problems like, this malarial vaccine example. But part of that will mean I'll be showing quite a bit. Let me see. I'll start sharing now an internet browser. Because I think that's the other nice thing about the tools that I picked. For those of you who saw kind of the syllabus, and just to double check, are folks seeing now an internet browser? Is that loaded correctly?
Yes, we can see that.
Perfect. Thank you for confirming. But anyway, I think part of why I chose the tools that I chose is, they don't cost any money. They don't require you to, have a particular license at your university. All of you right now should be able to access these tools with only an internet connection and use them for your research going forward on these projects. So I think actually the first tool that I might show is this UniProt database. So not everyone might have used this before, but I can kind of quickly orient to it. So it's, I think, really the best resource that exists for what's currently known about proteins. These can be proteins of the human body, proteins of the malaria parasite, any protein you might be studying. This is kind of the first place to look about it. It's going to integrate information from published papers. What we'll do is just kind of enter in the search bar the information about the protein that we know. So remember, Plasmodium is the name of this parasite we're trying to make a vaccine against. And then RH5 is the name of the protein. So you do a search. And it gets a set of results like this. I think part of the reason I chose this example as well is it also shows that even though, like, why is someone showing me how to do search in a search bar? It can actually go wrong, like an example here. This top result is not the correct protein. If you look at the name of it, Uniprot's sorting algorithm is not quite as smart as I think us people, but it found the name is called RH5 interacting protein. And its official name is RIPR, not, if you go to the next line down, the official name RH5. This is what RH5 stands for. So just like a little message I want to communicate there of, do double check what your amino acid sequence is, particularly for less studied proteins, it can be worth just making sure you have the right entry. Cool. And then it will have this identifier number, plus give information about the original species. So, Plasmodium falciparum is the main cause of malaria or, malaria symptoms in humans. So this looks right. Now, I won't go through all the, treasure trove of information that's on Uniprot, but, there's a lot here, I think, for you as you keep doing protein engineering. It will show you a nice little view of where it naturally is found. In this case, it's kind of secreted or on the surface of the parasite. It'll give you information about kind of what the protein's overall structure looks like, information which I'll actually talk about next workshop of what these signals mean and how to work with them. But I'm not going to address that this particular one. Plus some of these modifications to already tell you where there's modified areas, like this glycosylation at site 297, and a whole bunch more information. There's so much here. I'm just actually going to use this left panel to jump down to sequence, and then we'll see our amino acid sequence here. But in this form, it's quite hard to work with, as you can imagine. It's just a bunch of letters, hard to actually visualize. And so this is where our first tool of the series is going to come in, of benchling. So what I'm going to do is copy kind of the identifier code, which will start with a letter and then have, I think, 6 digits total. So I'm going to copy that. And then we're going to transition on to that benchling tool. And again, thank you to the organizers for doing a quick preface to how benchling works for those of you who attended. But if you missed it or any details like that, I'll at least help get you oriented to where you need to be. Let me just clean up my workspace a little bit. Close all those. So on benchling, you'll start out with a projects page that has kind of a list of projects over on the sidebar. You'll then, in my case, I'll just kind of navigate to the folder where I've put you for this particular project. And then also close out of this existing thing that I had. So if you're doing this for the assignment after this workshop, you should have a workspace like this. And you can, if you want, create a little folder for yourself to keep your files organized. Like, I'm going to make an RH5 fusion vaccine. And so now all the work that I'm doing about making this RH5 fusion vaccine will be organized into this nice folder. And you can actually kind of pull this up if that's helpful to, navigate backwards and forwards. But I think at the moment, I'll move that back down. So we want to add all this nice information Uniprot had, and benchling makes that very easy through this plus button import tool. So we go down to amino acid sequence, import from database, and then just type in the identifier I mentioned before. And once I do that, I didn't click the wrong button there. Try that again. And press import there. And there we go. Just a little bit of a delay. Now I imported it twice. Let me just delete that one or archive it. So anyway, now when I do that, you see benchling automatically created this really richly annotated protein sequence. This is, really great for us because it's going to have, I think, all the tools that we need to do our amino acid sequence analysis, plus more. So you saw it's already labeled this signal sequence that I mentioned I'll talk about next workshop. It's labeled the alpha helices, so we learn about secondary structure, where those elements are. It labels things like this disordered region, which you can see in this kind of gray color. So, there's already so much information that we got just by, pressing a few buttons. That's going to help us engineer this protein and do the next steps we want to do. So I think the first tool I want to show you is where these nice colorings are from. If you noticed, kind of you have these single letters for each amino acid, and right now they're actually being colored based on their chemical properties. And that's making it, me as kind of a trained user, and hopefully you after this workshop will be able to just kind of glance at all these spectrums of colors and already learn a lot about the protein. For example, my eye is drawn to this red region at the start where the signal is. The redder amino acids, again, you can use the codon table to help you remind you what amino acids which, but these are all quite hydrophobic, or at least there's a stretch of multiple hydrophobic amino acids right together. Over here, there's this quite bright bluish region. These are actually a highly charged region. If we look into this region, UniProt gave us this handy annotation. Region of interest, let me try that again. Region of interest mediates the interaction with human bathogen. And bathogen is the name of the protein on red blood cells in people that the parasite uses to infect. So again, I'm a bit more experienced, so I can kind of glance this just by the colors, but I just want to make the general message of once you're familiar with proteins, just by glancing at a color-coded schema like this, you can already learn a lot about, this region might be interior to the protein because, it has a stretch of hydrophobic residues. This region has, multiple helices in a row separated by electrostatic amino acids. There's just so much you can get just by this color scheme. So I'm going to show you how you can actually customize these colors so help you with your projects and to get this same kind of intuition. There's a little cog wheel you'll see in your benchling menu, and then coloring schemes. So you press this little wrench icon. Apologies, might be a bit slow, but hopefully it's working well. Cool. So right now it's colored by hydrophobicity, how much they like water or are repelled by water. We can do other colorings, like this RASMOL is even more detailed. You can see now we have doubled our color palette to all different kinds of shades. And now the positively charged and negatively charged will be red and blue respectively. We'll kind of have these greener shades in between for some of these. And special amino acids like proline will get their own color to help make them really stand out. So when I mentioned, be aware of where your cysteines are, where your prolines are, a coloring like this can help you do it. But I think for the moment, I'll actually just stick back with this hydrophobicity coloring. Just kind of want to make sure that you all know where that tool is. Now, particularly as you're getting first started, benchling has a nice additional functionality where you can draw little plots. So let me press this hydrophobicity plot here. And so now these bars, the height of them is how hydrophobic it is. So if your eyes aren't quite, as attuned quite yet to proteins and how they look, you can use these bars to help guide you. And now you can see what I mentioned a moment ago of this signal sequence having a really strongly hydrophobic region here. You can see this really highly charged region I pointed out to you near the binding site to that human receptor. And, all those features with time, you can train your eye to catch, but with these nice benchling tools, you can make them a lot more apparent. These last two here are kind of a bit antibody specific, but I will mention these lability sites as one last point. If we do that, it's going to add these small dots. And think of these dots as a bit of like a warning. Essentially, they're telling you the amino acid there might actually be modified in some way or look differently in the protein. So you can already get like a head start warning of, dealing with this region here, which is where the bathogen binding site is. There's, potential modifications that could be happening around where it is. Cool. All right. So I think from there, let me just check my notes to make sure I'm still on track. Yep. So first thing I'm going to want to do for us is to make sure that our vaccine that we're about to create does not bind onto blood cells. As I just told you, using our amino acid sequence, we've been kind of pinpointed right on to UniProt's annotation for where in the protein binds pathogen. I think more specifically, if I highlight my mouse over around here, hopefully this shows. Yep, this is a particular sugar site within that binding region. And right around kind of the critical part where this protein binds onto blood cells. I mentioned that we want to remove that functionality in our vaccine, right? We don't want our vaccine binding onto all the patient's blood cells. So a very simple tool that you can use in your research is what's called, alanine scanning. It's based on the principle that the amino acid alanine, or the letter A, is quite neutral. So lots of amino acids can be changed to A and not say, disrupt the protein entirely. So we don't want to totally break our RH5 protein. We just want to make it not bind blood. Let's kind of give like a short little example, but I might make a short scanning mutation around here, and you can read the papers actually in detail of the exact atoms where RH5 binds to human blood. But I can kind of simplify that and say, once you know the key region, which is identified here, you can change those amino acids to alanine. I'll just show in this quick example here. And so you can highlight residues in benchling and then type in. So I just typed in AAA, pressed enter, and now I've already sort of made my first modification to the protein sequence. And you'll always need to, I think, experimentally verify the changes that you make. But in this here, I'm kind of intending this change to make sure that my vaccine no longer binds onto blood. Something that I think is really important to do to remind you as you're making these modifications to a protein is to add what are called features. So to do that, select what you want to make a note. Right now, I want to make a note to remind myself, I've mutated this region to change the function of RH5. So highlight it, right click, whoops, or not that right click, and then you should see create annotation. And annotations, again, they're reminders, and there's ways that you can, share what you did with others or, just with yourself down the line. So I can say, mutated to alanine to remove blood binding. So there might be a slight delay, but hopefully that will appear on your screens. But, just gonna give it a somewhat memorable description. If you want to do more advanced, you can actually do like annotation types or, customize the color. Things like that. Apologies that this is, I think, struggling a little bit with the Zoom window running at the same time. But, if the annotation doesn't have time to work, I'll just move on to the next step. But you can hopefully get a sense, as we're making modifications to the protein, you can then use these tools like annotation and then change the protein. Cool. So we've already done kind of our first line of engineering. We've imported all this information on RH5. We've used it benchlink's amino acid sequence and coloring and graph tools to figure out some information about the amino acids themselves. And then one last thing before we move on to the next section that I also want to show is we can look at overall properties. So there's a tab here Benchling has called biochemical properties. So if I open this, it summarizes basically all the protein together or whatever part you highlighted. So right now it's just doing the entire protein, and it gives you information its total size. Isoelectric point essentially tells you at what pH is it neutral. Remember, lower pHs mean there's more protons. So if your pH is below the isoelectric point, it has more of H+, or it might be more positively charged. Kind of as a helpful way to remind yourself. And there's other little numbers here that can be handy as you're designing proteins. There's this number that I'll admit is somewhat approximate or rough, but can be useful as a general guide called the instability index that can help predict, if you're making lots of changes to your proteins, what are the odds that your changes are so large it might just totally disrupt it? In the 3D structure prediction tools I'll show you in a moment will also help you do that. Yep. Yeah, is there a hand there?
Yeah, hi Jared. Just before you move on to the other programs, there's two questions in the chat.
Yeah.
Yeah,
Yeah,
I can read them for
exit
you,
the
or can you?
sharing and quickly
Okay.
answer them. Yep. Would you mind reading them? I can
Oh, yes,
probably
yes.
do it
So
myself, but
Selma,
it might take
Selma
you.
asked, what if the protein you chose has missing residues? Do we use the same processes which you showed?
Yes, no, thank you. That is a very good point. And I think it's something I should have highlighted a bit more where, particularly in some of these databases where you have protein structures, they might miss parts in between. And so that might be kind of my understanding of the question, at least. So if you have missing parts of your protein, I think you have to kind of have a strategy going in. So one strategy might be if you are confident you can kind of modularize your protein, that the parts you know about, you kind of know their function, they're sort of modular domains, and you're just missing maybe the linkage between them. In that case, I would engineer it just as we're doing now. You might have like a modular domain of, this part, say, binds pathogen, binds this blood receptor. And, I can just modify these residues around here, and all it's going to do is change the binding to that blood receptor. Now, if those different parts of the protein in the missing parts in between are all, one cohesive unit where a change on one could very easily propagate and change the rest of the protein. That's definitely a much harder situation. And so I think you have to, at least my advice there would be, you would still use these kind of tools, but you'll want to complement them with a good understanding of the protein's biochemistry, and then information like if in the part that is known, if there's maybe an alpha helix towards the end, right around where the missing part starts, what's known about that region? Is it particularly mutation sensitive, a risk of disruption, or that kind of information? So it's a tough problem, but hopefully that kind of addresses part of it. Yep. And then what was the other? I'd be actually a few more questions now. You see, Erica, you have a hand up?
Yes, thank you so much. Please, can you hear me?
Oh, yep. Hear you loud and clear.
Okay. Yeah, so I have a question in the chat box, but I actually have another question. So the question in the chat box is that why did you mutate to alanine instead of like other proteins? And the is that in the protein structure, I see that there were more than one region of interest. So
Yep.
why were you mutating only one
Yes,
region?
no, both good questions, and I think somewhat related as well. Part of this is for the demonstration. I'm simplifying a little bit in the sense of I've worked with this protein in the past. I know a bit about it. So I think I'm no longer sharing, but out of those regions of interest, I kind of knew the answer ahead of time, and then could help guide you by mentioning like, oh, look at this positively charged region. It's quite unique within the protein structure, and that happened to point us right to the blood binding site. But if you don't have that prior understanding of your protein, my advice would generally just be investigate them all and kind of read up to understand what they're doing. It's a bit of an unfortunate answer because it does require a bit of extra work, but um, I think you're quite right to catch that because I was simplifying a bit from my experience. But if this is a protein that's new to you or you want to understand, you really should do your homework, so to speak, and know this region of interest has this property, this region of interest has that property, and then you can more confidently make the mutations. Now, about the alanine question, I think the simplest answer is if you look at kind of how all the amino acids look, alanine is a really nice point, sort of in the middle, where it's um, like not strongly hydrophobic, not strongly hydrophilic, not charged or anything unusual like that. It's, really nice in the middle. And so what I want to do with this mutation is I want to just prevent binding to blood, but I don't want to unfold the protein, right? If I unfold the protein, it might generate a poorer immune response or immune response against something that I don't intend because, unfolded proteins look different to the immune system, particularly, B cells and the antibodies they make. And so with alanine, I can hopefully replace those amino acids in a generally neutral way. But I will give you a kind of a preview of one of my bonus slides, which is, I don't have time to go to it right now, but there's actually kind of conversion rules that you can use to make other kinds of substitutions that are like alanine, likely not to disrupt the protein. So a famous one is called like this QTY code, which essentially means, if you see like a certain amino acid in your sequence, I think one of them is like a leucine in the sequence, you can easily convert it back and forth to one of these Q or T or Y amino acids. Let me see if we can pull it up. Yes, I think phenylalanine, I think is the example there. Phenylalanine can be converted to tyrosine quite safely, and it will change, say, the binding, but won't disrupt the folding. Leucine can be converted, what is it, to glutamine Q, the amino acid. But essentially the message there is I just don't want to disrupt the folding, but I do want to disrupt the binding. So I just want to make a neutral change that will disrupt the binding, but not the folding. And there's many ways of doing it. Alanine's kind of the simplest, but there's also more advanced rules like this QTY code. That will be in my bonus slides explained a little bit more because it's, I think, a bit more niche. Hopefully, Erica, that kind of answers that and how it looks. Thank you. Oh, yeah, and I saw one more, I think, that came up during the answering, or a couple. I won't hopefully take too much extra time for this because we'll have a chance at the end for more elaborate questions, but maybe one more if we can fit in. Yes, Pearlie, did you notice what that one was that came up?
Yes. So the question is from Helen. What if you're working with a novel protein? Does the design process stand?
Yes. So I'd maybe ask for clarification. Is that like when I mentioned the layers of protein engineering, like a de novo design protein that doesn't exist in nature, or like novel in what sense? I guess I'd need to know a little bit more about to answer that fully. I don't know if Helen's online and able to kind of clarify that, or if not, I can give kind of a general answer. So yeah, the more general answer would be if it's a novel protein, part of it is you can use your intuition to get a sense of important regions, but you'll probably have to make lots of versions. So right now in this workshop, I'm going to show you a nice clean example. We have a protein and we're going to make one version of the vaccine. But the thing also relates to the nice question we had at the start as well, of like uncertainty if parts of the protein are missing, how you deal with these situations. One of the main ways to deal with them, frankly, is just try multiple ways. If you have uncertainty of, well, this part of the protein is really novel, I don't understand it as well, or it's from an understudied species. It might just be in that case, use your kind of a creative intelligence to, make one design, and then the next day kind of revisit it, try a different design, what you're thinking of then, and then in the laboratory or using those tools, that will then be kind of the final judge of which design works. But certainly the more uncertainty you have, I think the more you might have to consider at least making more than one version. So I think it's really helpful, and thank you for those questions, because what I'm about to show you will look, quite neat and tidy. It's part of a demonstration, but in regular research, it might be, I'll use that really nice question about the alanines again. It might be I mutate this one region to alanine. Maybe I use the QTY code to mutate another region of that binding site, and try a few different mutations to make sure it's not binding blood. If I have uncertainty about which kind of modification will have the desired effect. So, the more uncertainty you have, the more open you should be to trying multiple things out. And, like kind of imagine on the bench link I was showing a moment ago, kind of duplicating your project and saying, here's one version where I made them all alanine. Here's one version where, I deleted them perhaps entirely, just kind of cut them out. Another version that's a different variant, and use your knowledge there of the protein to kind of guide those variants. Cool. All right, so I'll get back to some of the other questions that I might have missed towards the end, but let me just resume to make sure we stay relatively on time for the demonstration. Anyway, so we now have kind of the first layer of engineering done. We've imported the RH5 protein sequence that's going to be the basis of our vaccine. We've removed its blood binding function through our mutation. And we also explored, kind of regions of interest on the protein that are both annotated and that we can find through these tools like the amino acid sequence coloring and biochemical properties tab, which just one quick note is you can highlight, say, this region of interest here that's labeled as disordered by UniProt. You can highlight that, and the biochemical properties will now be only about that sequence. It can be quite helpful to tell you, this agrees, it has a very high instability index. Its isoelectric point is relatively low, and so you can learn a lot about the protein there, kind of from these different really simple interaction tools that might help guide your variants or how you design your protein. Now the next part I want to go over is how we're actually going to now make a fusion. So I said at the start of this that there's already a vaccine that had some success by fusing this RH5 protein from the malaria, or sorry, fusing a different protein from the malaria parasite onto an immunogenic protein. So we're going to try and achieve the same thing here, but the first question we'll run into is where on the protein do we make that fusion? There's several ways to do this, but I'm going to show you, I think, one of the more versatile ways, which is by predicting your protein structure using some of these nice tools like AlphaFold, and then from that structure, looking at it, inspecting it, and then having that guide where to make your fusion insertion. So what I'm going to do before I leave this page is going to highlight my amino acid sequence. Now, it's worth noting that this particular protein does have reasonably good experimental structures in the UniProt database, but for the purpose of this exercise, I'm actually going to predict it de novo partly because we've already modified it, right? Like it could be that this change here we made to remove the blood binding might change its structure. So, it's, a good motivating example, at least, of why we might want to use this AlphaFold algorithm. So there's a website, I think might have been linked out in the earlier notes, or that will be on my slides, called AlphaFold Server. Basically, this tool has become almost so ubiquitous, there's many options. If you're carefully eyed, you might have noticed benchling itself has this little button here that can predict structures, but benchling tool is a bit more limited, so I recommend that you go onto AlphaFold Server. If it's your first time, it will request that you just sign into a free account. So just like benchling, there should be no barriers to access. And then how it works is quite simple. There's a little box here. I've already filled out this box, so I'm going to clear it. And then in this, you can paste in your modified amino acid sequence. So now here's our protein with that little modification that we made to remove blood binding. And kind of taking this and trying to predict what's its full structure. So you can then continue and preview job. It'll just give you a confirmation. And what's quite nice is you'll get 20 of these for free per day of structure predictions that are quite fast. If you're doing a really intensive project, in my bonus slides, I give you a link to a free kind of version of this called ColabFold, where you can run as many of these as you want. You can customize it, but it's a little bit more complex to use. It requires, some knowledge of programming and entering the notebook. So I think this is kind of your go-to tool. It's also the most up-to-date. ColabFold's gone a little bit out of date. So we'll press confirm and submit job. Now, depending on how difficult of a job you just gave AlphaFold, this can take on the order of maybe 10 minutes. So conveniently, I've already kind of like a cooking show. I have a pre-made the protein structure prediction. So imagine we waited the full 10 minutes. AlphaFold would spit out something that looks like this. So on the prediction, I think there's a few things I want to highlight, and then we're actually going to dig deep into the structure. So don't feel bad if you don't kind of get all the nuances down yet. This is just like a summary, and then we're about to do the structure next. So AlphaFold is going to give you this view, which is going to be your protein structure, nicely colored based on the model's confidence. And it already groups them up into categories. So where you're seeing those yellows, and especially the oranges, you should have a lot of caution of were they correct? Or, as I was saying during the introduction slides, it could be, for example, that some of these are correct, they're just disordered regions. So AlphaFold will always give a low confidence score to something that, doesn't look at all like a nice structured region. That's maybe kind of like one of these floating loops that you see here. And so, take that into your consideration that particularly if you know your protein's disordered, if you remember our UniProt annotation kindly, gave us a few places that are known to be disordered. So we know this is a property of our protein in some places. UniProt will highlight those orange, but they could be correct. And conversely, something like this yellow helix here may or may not be real. And so it's worth kind of absorbing before we move on to the next step, just what the overall prediction accuracy looks like. You can see there's this very high confidence core. And hopefully now you can see a real example of what I mentioned during the intro slides of what these prediction tools are really good at is like the orientation of secondary structures. So, there's a whole bunch of these helices wrapped together in this orientation. You can say that very confidently. But then things along the periphery, things outside of the secondary structures, it struggles with a bit more. This is just kind of like a graphical representation of that structure of what's nearby each other. So you can get a sense of like longer range effects. Say if you modify part of the protein around here, it could modify the protein around residue 150. That effect could permeate all the way to residue 300, for example. So this is another thing to keep in mind, both for fusions and other modifications that you make. The structure itself, I think, is one of the key guides you'll need to use. But little plots like this can be handy as a reminder of, if I made a big mutation of amino acid 200 right around here, that could permeate all the way up to almost amino acid 300, affect those structures downstream because they're quite interlinked, nearby each other. So when you do this yourself, during the assignment or during your research, my recommendation is you just press this download button, and then AlphaFold will give you a zip folder. I think part of the reason I recommended this one to you is AlphaFold gives you not only the top prediction, which is what we're seeing right now, but multiple alternate predictions. So there's that really nice question a moment ago of like a hard to predict structure or a really novel protein. In that case, I might look at multiple of the predictions that AlphaFold gives you. Just double check how close they are to each other. And if they're really divergent, you should kind of take that as a bit of a warning sign. Or if there's a region that they consistently predict, you can be quite confident that's a real region. So I'm going to now show the next tool. So AlphaFold, its job is done. It's given us a 3D structure prediction of the protein and its confidence scores. So now we're ready to inspect the structure in detail. And so for that, I go to, again, its link should be available to you. The Royal Society of Chemistry has this data bank called PDB that hopefully some of you might have heard of, which is just the place that experimental scientists put their protein structures in, once they've done that hard work to figure out what the structure is, they put it in this database. But not only is it a database, but has this really nice viewing tool. So I'm going to show you how to use that because it's useful not only for structures you generate yourself through AlphaFold, but also if you ever use the database for, experimental structures that have been published. So once you get on this page, which again, we'll have a link to, you just navigate to import open files, select this, go to where you downloaded that folder. So in this case, sorry, I did several of these, but it will look like this. So if you unzip that folder that AlphaFold gives you, the numbers here are, when I mentioned it gives you multiple structures. And so 0 is the first one it does, and then 1, 2, 3, 4. For the purpose of this exercise, I'll keep it simple and only give the first prediction. But certainly for your own projects, it's worth just checking how similar they are to that idea of making variants, perhaps. Does one structure give you a different idea, etc. Then you press apply once you've kind of selected your model. And then here we are. So it looks just like what we're seeing in AlphaFold's preview. But now that we're in a proper tool, there's a lot more we can do with this. I think the first thing that's very helpful is there's a interactive map of the amino acids here. And hopefully you can actually see if you look closely. As I'm moving across, it highlights them in real time. So you can see where I am on the protein structure. Here I am on this loop. This is the N terminus, and then it's kind of winding down across. And with this little mouse button here, I can make selections. So just by eye, hopefully I can get this correct, but here's this highly charged region that we saw earlier on benchling and that I mutated. So let me get that highlighted. And then, so I think this version doesn't have my alanine mutation because it was an earlier one that I did, but hopefully it does the same job. And so we can see right around where this binding pocket might be for where this protein binds the blood receptor. And so hopefully you can see this now in light green. If you want more complex functionality, there's these, I won't go into too much detail, but there's what's called components that you can add. So I can add one to the current selection that adds, for example, ball and stick structures, just like you saw in chemistry class. I'll press create. And if I zoom back up, look, it's now kind of given a fuller picture of this binding region where RH5 binds the blood receptor. So it can be a lot of information, particularly as you're starting out, but as you get more familiar, you can get a sense of, for example, off the helix, you can notice how all these side chains, are branching out in that oriented fashion that's stereotypical of how helixes look. And then you can notice other amino acids kind of projecting out from this key region here that might be important to track. But, right now I'm doing this mostly just to kind of raise our awareness of this. You can assign this a different color if you want, if this is a region of interest you really want to track. You can really interact with your protein a lot more. So that was kind of how you do selections with your mouse, how you can add some basic layers of annotations, which again is by these components. So right now we have the cartoon representation for most of it. And then on our selection, we have this ball and stick. And if I want, I could even set the coloring differently. So I can do the same kind of things as benchling, as coloring by chemical properties. So residue property here, I can have color by hydrophobicity and do all kinds of functionality. We don't need to go into them right now because thankfully benchling has already told us some of the high level messages we need to remember. But at least I want you to be aware that there's a lot of functionality here, and now you have these kind of nice colors telling you where some of the charged regions are, which is what you're seeing in those bright colors, where the uncharged regions are of this particular functional binding site. Cool. So let's get back to the main goal that we set for ourselves. So now we've predicted the structure of this protein we want to make into our vaccine. But next, our goal is to figure out where to fuse this onto that immunogen that's used in the current malaria vaccine. So on the slides earlier, I kind of give you a brief walkthrough, but there's, I think, generally 3 steps. First is check the N-terminus, which is kind of where the protein starts. So back in our view here, that's these amino acids, N-terminus. Check that. Check the C-terminus where it ends. These are really nice spots because, as you can kind of see in my examples, if you fuse something on here, by definition, you're only, say, disrupting the part that it's attached to. Whereas if it's in the middle of the sequence, you're kind of having to insert it between 2 regions and you have to not disrupt either of them. I think that's the place to start. Look at your termini. See, do they look like reasonable positions to make a fusion? If not, or maybe if you want an additional variant to try in the lab, then you can look at if there's a safe place that could suggest itself outside of the key functional regions you don't want to disrupt. A good kind of informal way of describing it is like some loops, perhaps, that are accessible. So as I move my camera like this might look like an attractive loop. So if you're a beginner, you might be tempted to say, oh, this is a great place to fuse my protein. But that's where I kind of brought in our earlier annotations for benchling and moved the colors onto here to point out that this is our key binding site for the protein to be functional. And it's also quite close to the secondary structure. So it might be a dangerous place to modify too heavily. Or, it wouldn't be my first choice. You could try it, but it might fail. Instead, if I were to do an internal loop, maybe something like this could stand out to me. If you kind of see this loop over here, it's reasonably far away from the secondary structure. It's, has enough amino acids in between to kind of serve as linkers. And you can also create artificial linkers, which I'll mention in a second, to kind of give you more spacing. But, that could be a reasonable place you can imagine inserting a domain and not disrupting this delicate protein below. Now, for my actual fusion, I think my favorite spot, though, is not this loop here that I just highlighted, but it's actually probably going to be the C-terminus. Like I said, if you can get a good terminus, that tends to be one of the safest spots. And the C-terminus is quite nice in that it's projecting away from my protein. So imagine when I gave you that slide about intuition, these proteins are moving at 50 kilometers an hour, bumping into each other. This thing will be flopping about in every direction. And so it's quite removed from that core, just kind of flopping in the periphery. If I kind of imagine this moving, not just in 3 dimensions, but a moving 3 dimensions. And at least this immediate ending part has no clear structure. To double check, though, it's always worth going back to your benchling annotations. So use these tools together. They're not in isolation. And if I go back to my benchling annotation, again, there's no warning signs that I see. It's all no particular features. It's not like, you notice here there's this disulfide bond region, which is what I mentioned is that key cysteine bridge between amino acids. So I would be nervous about modifying around here, but the C-terminus looks quite safe. Hopefully you can kind of follow the logic there. And hopefully you also see what I mentioned at my earlier slide of this is partly art and partly science. This is not a hard and fast rule. I put this into an algorithm. It says fuse here. It's, using our knowledge of protein chemistry, we found kind of 2 potentially reasonable sites around there, in there. And we used our combination of tools like AlphaFold, like benchling, and like this tool here to confirm that these are good sites to use. So with that, I think I've gotten the message that I'm looking for, which is if I'm going to make a fusion, it shouldn't be to the N-terminus. Right? Remember, the N-terminus is this helical structure over here. It's also related to the signal sequence of the protein, which I'll mention next workshop. You don't really want to mess around with without being very careful. So I don't want to modify that. This loop is okay, but I think I can do better by doing this C-terminus for my fusion location. So just to wrap up in the last few minutes that we have, I'll now implement this in benchling. So I got the information again that I wanted. I predicted the structure, predicted where it'd be a safest fusion site. So now back in benchling, using this. Let me just navigate quickly out. In one of my folders, I have downloaded the immunogen part. Here we go. of that existing malaria vaccine. Just take a second to load. Here it is. What's funnily enough is it's actually quite understudied, just in terms of not having as many of those rich annotations as RH5 did. So it's a really nice example, and I love the question earlier about working with a new protein or one that's less understood. I have this chemical coloring already just because I know it's going to be quite important to double check how this looks and make sure that as we make this fusion with a somewhat less understood domain onto a malarial protein, that it all works together. But just to kind of show the simple version of this is now I have my 2 amino acid sequences. I have my RH5 that I've mutated to remove flood binding. And then I have this immunogen that makes the existing malaria vaccine more potent. So I'm going to just simply select all of this from my immunogen part, copy it, go back to the RH5 protein. Remember where we selected for our fusion site was just the C terminus, so it's easy to find. If you wanted to do this loop, I think one of the easiest things is you can kind of use some of these selection tools. And so notice what just highlighted in pink. It was in this case a glutamine around position 280. And so now it's an easy way, highlighted in green there, to orient yourself if you're jumping back and forth between tools. So where is it? If I can hopefully have this work in real time. Yep, glutamine right around position 280. So that was the first loop that we found. But you notice it's in this kind of disordered region. It has a disulfide around it. So it's not my first choice, but it might work. Anyway, so back to where my first choice actually is. It's the C terminus right here at the end. Sorry about that. It's being a bit slow, but I'll give it a minute to load. Here we go. So I just copied the sequence of that immunogen here. And so I'm just going to copy and paste. The BenchLink supports that. Press enter. And now it's put onto the end of my sequence, this immunogen. So I'm going to go back to, if you remember how to do an annotation, I'm going to right click. Hopefully it doesn't lag my computer this time, but I'll try this again. Immunogen HBS AG. I'm not sure why this is going so slow, but hopefully you have better luck on your own. Yeah, it's starting to work. Anyway, so I'm just adding an annotation to remind myself. Right now it's a bit complex. We have a mutated RH5. We have this part of the RTSS existing malaria vaccine kind of all together. So I'm going to want to save this annotation to remind me where this is. And then as kind of a last note, if we go back to our structure, I think as a good rule of thumb, you can see that we are a bit removed from this primary, kind of primary structure of this, kind of this really nicely ordered core. But if we count our amino acids, so I just press the selection tool to help me count how much there is. What is this? It's only about 6 or so amino acids removed from this really delicately structured region. And so I'd be fairly nervous about attaching a really big fusion so close to my structured core. So some of you might have heard this concept of linkers before. So we're actually going to try one of those here to add a little bit more distance between our vaccine protein and our fusion to make it more immunogenic. I'm going to add a small linker. Now in the bonus slides at the end, I actually give you a set of common linkers that you can choose. So there's some nice ones, like some that are deliberately in alpha helix, which might sound a little bit counterintuitive, but if you remember, alpha helixes can be kind of like nice rigid rods. It's kind of an informal way to think about them. And so, a nice rod linker could be a sensible choice. Or you can do what are called flexible linkers. I think the most famous is it's called GGS, or some people put 4 Gs, but several of these glycines that again don't really have a side chain on them. They're quite neutral, interspersed with a few serines. And serines is also fairly neutral, but has a bit of polarity to help it kind of wiggle around a bit more, so to speak. Polar amino acids are on the water surface, so they're going to be more flexible in moving in, whereas a hydrophobic amino acid, let's say like a W, which is tryptophan, is going to want to kind of avoid water and might kind of bury itself in your protein, among other issues. So I wouldn't choose it for our linker here. We had about 6 amino acids, if you remember, on our structure before, separating us from the core. If I added just GGS, that would bring that up to around 10. But I think I'll maybe duplicate that to have another GGS repeat and use this as a nice, simple, flexible linker. And so in the bonus slides that I have at the end, and maybe if folks are interested in sticking around, I can kind of give a brief preview of them. But I give a few options for linkers in situations where you might choose them or might prefer one over the other. But for this simple example today, I think that suffices. So I've kind of recap all that we've done because it was quite a lot. We went to the UniProt database, got information about this malarial protein. We imported it into Benchling and used color and annotation tools to learn about its structure, highlight important regions, and also make our mutation to prevent blood binding. Then we used AlphaFold to do a computational prediction of what that mutated protein structure might look like. Keeping in mind some of these caveats in the prediction, and the fact that AlphaFold is traditionally not strong at predicting effects of small mutations like we made. But we had at least a full structure to work with to inspect in detail, find our preferred site for making the fusion, which in this case was the C-terminus. And then I went back to Benchling, fused on this vaccine portion to our C-terminus, added a short linker, and there'll be a bonus slide that gives it a little bit more nuance there, and kind of putting it all together as this would be my proposal for how to make this vaccine. I'd reiterate that fantastic question from earlier that, there's difficulty here in, predicting some of these domains. Some of these proteins are, not as well studied as we might like. And so it could be, if I was doing this in the laboratory, I'd make a few other versions. But, if kind of as a first starting point, I would say this is a intelligently designed, engineered vaccine for malaria, combining these two different protein domains. Again, we mutated the function of one, and then we've kind of fused on the function of another through that little linker sequence. And, all together, I think is a reasonable thing that you can then try in your experiments going forward. Now, I won't kind of show it for today, but you can then actually iterate on this. So you can, if you saw what I just did there, I just highlighted everything and then copied my full sequence. And then you can go back into tools like AlphaFold, or in my bonus slides actually give you a number of different prediction tools, because even some of the older fashioned ones can be just as good or better in some cases than newer tools like AlphaFold. And iterate. So I made my fusion protein. This is the original. Now we've added this fusion. And so I can now submit this job to AlphaFold and have it predict what does it think the fusion looks like in 3 dimensions. And then, if the fusion suggested a problem, so I'd compare it then to this structure. If it destroyed, part of this internal core or disrupted the key functional regions of the protein, then I might iterate and say, oh, maybe I need a longer linker, a different kind of linker, or maybe I need to fuse this in a different location. So, for time purposes, I won't, go through the iteration right now. But hopefully you can, see how it's quite easy to do that. This has been half an hour or so. I paused to answer questions, so, not too much time. But in that time, we made a reasonable design, and we're now at a step where you can, repredict the structure by AlphaFold, see if that structure looks good, and then maybe modify this design if we notice anything amiss or that we want to change. So I think that was, I'll just double check my notes, but I believe that was everything that I was hoping to get across in the demonstration. So again, hopefully you kind of feel like you have a good toolkit now at your disposal of, structure prediction tools, structure analysis, primary amino acid sequence annotation and visualization tools, and just some of the concepts that you might go through designing these kinds of mutated or fusion proteins, the practical tools that you need to get you there. So as maybe a bonus, I can stick around a little bit after this main session is ended for any follow-up questions. But I think if not, that was all that I had to present today, and we'll kind of circulate out an assignment where you can practice some of these and gain more experience and familiarity. So again, that was kind of all the formal presentation. So I'll switch on to questions. And for those of you who might be pressed for time, or at least some of you are in quite far away time zones, do feel free to take a nap if you don't have any questions right now. But happy to stick around. I see Shakira, you have a hand raised.
Oh, yeah, thank you very much, Jared. So we're just here to read out some of the questions in the chat.
Oh yeah, very helpful. Thank you.
From the previous question, there was, how can I determine if my mutation affects the protein's function or not? That was from Fatima Zahar.
Yes. So I think part of this is understanding where that function comes from. So there's really good examples in both binding proteins and enzymes. Our example here was a binding protein, and I didn't show it today, but there's a published paper where you can actually see the at an atomic resolution what atoms are kind of binding each other so that this malarial protein can bind pathogen. And if you trace those atoms, you can find what side chains of the protein they belong to. And then from that, I can be quite confident if I just mutate those few side chains to something neutral like alanine, related to that earlier discussion that we had, I'm going to remove the points of contact that are needed for that function to happen. Enzymes are another great example. There's, certain known mechanisms like serine catalysis where you can be quite confident if you get rid of that serine, the enzyme won't function, or, there's kind of predictable ways to modify that particular class of function. But it will be protein dependent. So if you're working with a new kind of protein that's you don't really have the binding sites mapped out as much, then that's back to, I guess, my earlier point of you might need several variants to know exactly, what do I change the amino acid to? Is it alanine? Is it something else? Which amino acids do I change? How many do I change? Those questions become harder if you have less information. That's kind of the approach.
All right. Next question from Haz. How do you ensure that? Sorry. How do you ensure that the mutated protein retains its stability?
Another good question. So I showed a couple example tools so far of, honestly, I think the best that we can do to predict that without doing experiments, just if all you have is a computer with you. So one was calculated metrics like Benchling stability metric. If you make your mutation or fusion, and then all of a sudden you're getting really high predicted instability through that tool Benchling has, it's at least a warning sign that you might, have made too much of a change or it might have disrupted the protein. Another is through those 3D structure predictions. So it's not a hard and fast rule, but it's likely that if you take a protein that it's in its wild type state, like I did with you can do with RH5 as an exercise, you just put wild type RH5 through the prediction, and you get a nice clear prediction of that core that I showed you in the 3D structure of those helices. If then you make your modification, in that same algorithm is now struggling to find the structure, or is the core looks very different, then that's again a pretty likely warning sign you might have disrupted the protein function. Now, in terms of computational tools, I would say that's again about as good as we currently have. Even the best engineer, you might make a modification that looks fine. The predictions still look good, your stability indexes still look good. There's nothing obviously wrong, like you broke a disulfide that was essential for the folding, but it could still have an unexpected effect that breaks the function or does something like that. So, always be prepared that that will happen. But with these prediction tools, at least you can reduce the probability it will happen.
Okay, next question from Teddy. I think, I don't know whether you talk about this in the last session or but she's asking for an example from food application, I think.
Oh,
Yeah.
food example. I see. I think one of the first ones that comes to mind is I think a lot of cheese making will actually rely on an enzyme. If I remember correctly, I think it's called renin. That's part of what will actually make the cheese form when you kind of mix the way in the ingredients. And a lot of that will be kind of a recombinantly produced protein that you'll produce, mix in with your batch. I think the other famous example that's getting more attention now is ways that you can replace certain foods that are currently in use, most particularly around like meat products that might require killing animals with proteins produced, designed, that you designed, put into cells to produce, say, like mimic a muscle fiber in a dish, that kind of protein. And there's a lot of really cool research, at least over in Cambridge where I am, of, ways that you might add flavor by protein engineering and, you kind of affect how the protein interacts with your tongue and your taste receptors, or, examples like that are quite neat. So that's a small example or set of examples to illustrate where it can be used.
Okay, next question from Salma. Some proteins need an extra something like a cofactor or an anion for them to take shape that is active or have an appropriate structure. In that case, how do we take that into consideration?
Yes. No, thank you, Salma, for that. It's I think an important point to highlight because not all proteins will be as easy as the demonstration cases. And I think proteins that require, these cofactors are a good example. I think the first place that I would go to is if, you're saying that you already kind of know iron is needed for that protein to fold, that knowledge might come from or might be related to a structure that's in the database. I don't know if I'll have time to quickly show you this now, but on that same PDB database where we viewed the 3D structure, you can view experimental structures. And so if you have something that is known to have that binding cofactor, you might be able to see the experiment that found that. And it may even be that you don't need to predict that structure as your starting point. You can just use that one that has the cofactor in place. And it makes it very easy to, be aware, okay, this is my cofactor binding site. I want to be cautious around manipulating this, or, want to avoid any changes there that you can incorporate. I think a future looking second part of this answer I can give is that these prediction algorithms are advancing very rapidly. So there's a published paper, but not yet quite translated to a tool yet that all of us can use on our daily basis that makes these predictions of cofactors and, is there a potential small molecule binding site on your protein? And, it could very well be at the pace that this is going in 2 years' time or 3 years' time, you press it into AlphaFold or the next generation folding tool, and it will incorporate those cofactors. But until we get to that future time, again, I'd look at, is there a known structure or maybe a homologous structure? So, if it's a hemoglobin-like protein that you're working with that doesn't have a structure itself, but it resembles hemoglobin, then you can maybe do what's called like homology matching, which you say what part of your protein looks like hemoglobin's iron binding site. And then you can make a good guess that in your protein, that might be its iron binding site. And was there anything else? I saw a hand a bit earlier, but it might have been addressed.
Charlene asked, I think, I'm not sure, but I think it was answered. How do you know if a mutation will disrupt binding but non-folding?
Yeah.
Oh, yeah. Binding but not folding. Yeah.
Oh, yep. No, I think we had a similar question. Yeah, it was answered. Thank you. Yep. Yeah, but it is an important question, definitely. So, worth thinking about.
Okay. Well, I think that's all.
Very good.
Yeah.
And, by all means, I think part of why we structured the workshop this way is you have, another couple of days to reflect on the material. If you have time, maybe get started on the assignments. And then on day 3, you can also ask additional questions. I think there's a good amount of engagement kind of already on some of the concepts, and hopefully I could clarify some of those points there. And there's the assignment right there, just on time. Thank you.
Yes. Thank you very much, Jared. Thank you, everyone, for attending and being patient. I know it's quite late in various countries, but thank you for your patience. And thank you so much, Jared, for dedicating your time. Thank you, Presley, for sharing the assignment. Feel free to, I don't know, fill in that. Like, if you have any questions, any further questions, we'll be happy to respond to them. Thank you so much.
Yeah, thank you. And I guess I'll hopefully see many of you tomorrow.
Yeah. Freddie, over to you.
Okay.
Yes, I just quickly wanted to let the participants know, in the assignment, there is 5 feedback questions. So you can also ask a questions, anything that you were unsure about, and then Jared can see that question, and maybe hopefully tomorrow you can answer that if there's time. So we really do encourage you to please do most of the assignment if you can and have time before the next session. But I just also want to say thank you so much, Jared. This was really, really informative, and I really enjoyed it. Thanks.
Thank you. Very good. Yeah, we will see folks tomorrow, and a good night if it's a night in your time zone. And see you later.
See you all tomorrow. Bye.
Bye.
Thank you. Bye.
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 See you all tomorrow.
Okay, see you tomorrow.